The barrel eye, lot. Macropinna microstoma is a species of deep-sea rayfin fish, the only representative of the genus Macropinna from the family Opisthoproctidae. They have a transparent head through which they can see with their tubular eyes. The head through which the fish observes prey helps to protect the eyes. The discovery and initial description of the species was made in 1939 by Wilbert Chapman, but the first photographs of live fish were taken only in 2004. It lives at very great depths, so little has been studied. In particular, the principle of fish vision was not entirely clear. It was supposed that she must experience very great difficulties in view of the fact that she sees only upwards. Only in 2009 was it possible to fully study the structure of the eye of this fish. Apparently, when trying to study it earlier, the fish simply could not stand the changes in pressure. Macropinna microstoma is a relatively small fish. The largest known specimens reach about 15 centimeters in length. The body is covered with large dark scales. The fins are large, wide and rounded. This species is characterized by a long intestine with numerous blind outgrowths, a wide pharynx and a narrow mouth, which is the main limitation on the size of prey suitable for consumption. The most notable feature of this species is the transparent, domed shell that covers its head from above and to the sides, and the large, usually upward-pointing, cylindrical eyes found underneath this shell. A dense and elastic covering shell is attached to the scales of the back and sides at the back, and on the sides to wide and transparent periocular bones, which protect the organs of vision. This integumentary structure is usually lost, or at least severely damaged, when the fish are brought to the surface by trawls and nets, so its existence was not known until recently. Under the integumentary shell there is a chamber filled with a transparent liquid, in which, in fact, the fish's eyes are located. The eyes of live fish are bright green. The eyes are separated by a thin bony septum, which, moving back, expands and contains the brain. In front of each eye, but behind the mouth, is a large, rounded pocket containing a rosette of olfactory receptors. That is, what at first glance in photographs of live fish seems to be eyes, is actually an organ of smell. The green color of the eyes of this fish is due to the presence of a specific yellow pigment in them. It is believed that this pigment provides a special filtering of light coming from above and reduces its brightness, which allows the fish to distinguish the bioluminescence of potential prey. Off the west coast of Canada and the United States, south to the Gulf of California, Mexico, fish in their normal habitat are usually immobile or move slowly in a horizontal position. All fins are fully extended. The pectoral fins are oriented horizontally, and the pelvic fins are inclined downward at an angle of approximately 30. Due to the large open fins, the fish reaches a stabilized position in the water column. When the fish moves, all fins remain fully extended, and movement is provided by the caudal fin. In case of danger, the fish presses the pectoral and ventral fins to the body, and makes sharp jerks with the beating of the tail. Due to the special structure of the eye muscles, these fish are able to move their cylindrical eyes from the vertical position, in which they are usually located, to the horizontal, when they are directed forward. In this case, the mouth is in the field of view, which provides an opportunity to capture prey. Such features of the visual apparatus make possible two fundamental approaches to the search for food. First, the fish is able, being in a horizontal position, with eyes directed upwards, to notice potential prey above it. As the fish moves its body upward, with its mouth toward the prey to grab it, the eyes move forward, keeping the prey in sight. Secondly, with a constant horizontal position of the body, the fish is able to follow potential food objects that descend from above by moving their eyes. When a food object hits the level of the mouth, the fish grabs it. In the digestive system of fish of this species, zooplankton of various sizes were found, including small cnidarians and crustaceans, as well as siphonophore tentacles along with cnidocytes. Given that zooplankton accumulates in large numbers on the tentacles of siphonophores, where it can be preyed upon by deep-sea fish, as well as on the remains of cnidarians found in the digestive system of Macropinna microstoma, we can conclude that the continuous transparent membrane above the eyes of this species has evolved as a way of protection against cnidocytes of cnidarians, especially siphonophore.